Hi, everyone. You're like the only class I've ever had that waves like that. That's amazing. That's fantastic.
It's a low bar.
No, I'm not saying like, I'm saying like my whole life. It's a much higher bar. Okay, so some general pieces of information. First of all, remember that the clinical immunology assignment is due today. Lab report is due on Thursday. If there are any issues, please send me an email and let me know. Because apparently there was confusion about that. So let me know if there are issues. And in lab today, in lab on Thursday, we're going to do some data analysis of the data we did with adaptive immunity, but we're also going to do a fun activity. So I'm looking forward to it. And let's see here. That's that information. Most likely, the review session for the final exam, just so you know, is going to be on December 11th, which is on the Saturday. Your exam's on a Monday, just because I ended up having plans on Sunday. So it will probably be on Saturday, so just to be aware. Yeah. One other final piece of just sort of general info to point out to you is that if you think back to some of the past lectures, and this will, I'm sure, happen again, sometimes I tell you a little bit about certain clinical conditions or certain disease states. By and large, when I'm talking about those, it is coming from a bunch of years of experience of talking about them in front of students, where in many cases I have needed to make sure people were hyped up about the importance of this stuff in immunology. You guys are a fabulous class who do not really need any hype. But know that in general, for example, if I'm going on about how gross some condition is, that is not a lack of empathy. That is me trying to get attention. And make you sort of think about how we can work on patients. And so I hope that no one thinks that I am heartless about any of these patients as if I talk about the negatives of any of these disease states. If anything, I'm talking about it because I want us all to realize that we should actually do our best with our knowledge of the science to do something about it. Okay. I have a couple of slides here about autoimmunity that I didn't get to last time. I actually had a bunch that I didn't get to last time. Some of them I've put here at the beginning before I transition into our next topic. The others I have later at the end, so hopefully this will make a little bit of sense. But there were a couple of big picture points about autoimmunity that I wanted to make before I transition into allergy. So if you recall, we spent the last couple of class periods talking about self-tolerance and ways that self-tolerance, particularly peripheral tolerance, can fail. We also saw some ways that can often be related to some kind of infection or other environmental influence that can change immune regulation. You can also see, as is listed here, that there are some genetic factors that lead to some aspects of genetic susceptibility. We do know a little bit about some aspects of genetic susceptibility to autoimmunity. The most frequently associated or the most heavily associated gene with different autoimmune disorders is MHC. And so in general, in all of these cases, we're seeing some particular type of MHC that is presenting a peptide that is allowing for this autoimmune reaction to happen. You can see that in some cases, there is a pretty dramatic difference. And so in ankylosing spondylitis, there is more than 150-fold increased risk in patients with HLA-B27. In birdshot chorioretinopathy, you can see it's more than 50-fold for HLA-A29. You can see that they do get much lower In fact, you can see for type 1 diabetes, it's actually you are less likely to have DQ6. So there's a pretty big range here, but by and large, the biggest contributor genetically that we've ever really found has always been MHC. There have been a few cases where people have done some pretty massive genetic association studies to try to find genes associated with different autoimmune diseases, and they'll spend millions of dollars and they'll end up coming up with MHC. And usually my response to reading that paper is like, thank you, Captain Obvious. So this is sort of the biggest gene, although there are some other genes that are associated with other types of autoimmune diseases. And by and large, those genes are types of genes that you might expect. So you can see that many different types of autoimmune diseases are related to polymorphisms in genes that are related to things like antigen processing and presentation. So for example, you can see ERAP shows up here. That's one of the peptidases that cleaves a peptide to make sure that it can fit onto MHC class 1. You can also see that there are differences or other kinds of polymorphisms in co-stimulatory molecules or adhesion molecules. You can see that there are changes in cytokines or cytokine receptors or in innate immune response proteins. And so by and large, the genetic susceptibility piece to this has to do with some immune genes as well. There are some cases where there are some relationships with other genes that are not immune related. For example, in psoriasis, which is a skin-related autoimmune disorder, one of the polymorphisms that comes up is a gene that is expressed in skin. So it makes the skin more susceptible to being destroyed. So usually kind of that's where some of this goes with genetics. The one other piece to this that I want to point out, I've already told you about a bit in lab, has to do with the aspects of sex bias and autoimmunity. And you can see a graph on the left, and it shows you the relative rates of many of these different diseases in females versus males. If they are more prevalent in females, they're in pink, and the amount of sort of the increase in the ratio is given by the size of the bar. And you can also see this ratio in this table on the right. And so what you should notice from the majority of these diseases is that the majority of autoimmune diseases are dramatically biased towards women. And so most autoimmune diseases tend to be seen in women more frequently than in men. And so you can see, for example, Hashimoto's thyroiditis is 19 times more common in women than in men. And you can also see the other graph starts with Addison's disease being about 14 times more common. And so you can see that there's this big bias in almost all of these autoimmune diseases towards women. There have been a lot of hypotheses about why this is. So this is, you can sort of see, I'm somewhat tying this with some aspects of genetics here. And one of the things that people have often talked about is that many of these autoimmune diseases, some of the thyroid diseases, lupus, some of these others, tend to show up in women after their childbearing years. When a woman goes through pregnancy, there are some massive changes to her immune system so as to not reject the fetus. And so there has been this idea that the changes to your immune system having to change and go back and change and go back during pregnancy somehow does something that may predispose you to autoimmune disease. So it's sort of an effect of pregnancy. There's also some idea that different sex hormones do directly act on immune cells, and it's well known that a number of those sex hormones do directly act. In fact, many hormones act directly on immune cells. We'll talk more about one of those as we get more into allergies. But something that is somewhat related to some of the sex hormones is cortisol, which is a major stress hormone, because when you're stressed, it also impacts your immune system. It's the same kind of thing. And so the other question is whether some of the sex hormones may be impacting immune responses. And as I've told you before, we do in fact see a general difference in immune responses between men and women, whereas in females we tend to see, say, a higher general T cell response. In males we have a lower response. That means with males we may have viruses sticking around and causing more damage because they don't have as good a defense, whereas in females we may have more immunopathology. And so this may also underlie what's going on with differences in autoimmune disease predisposition. And you can see some of the differences in viral infection between men and women. On the left, again, we've already talked about some of this. We have also realized that in some cases, some particularly important immune genes are encoded on the X chromosome, and many of them escape X inactivation. And so, in fact, there is a difference in gene dose in men and women of those genes. Women actually have 2 copies and men have 1 copy. And so that may also change immune responsiveness as well. And that's sort of something that is a bit newer to the field as well. So those are kind of some of the things that are related to the other side of this. It's not just environmental insult. We also have all of these kind of genetic factors. This table shows some common autoimmune diseases. The reason why I'm showing you this table is not because I'm going to go through all sorts of details of many of these autoimmune diseases. The reason why I'm showing you this table right now is to show you that we can often classify our autoimmune diseases as being either antibody mediated or T cell mediated. In addition, we see some are more systemic, and that's especially true of those that are affecting central tolerance, though there are some that affect peripheral tolerance that are also systemic, as well as some that are organ specific. And so what I want you to see here is the way that we often classify some of these autoimmune diseases as being an antibody disease or a T cell disease or something like that. Yeah, Jamie.
When it says immune complexes,
It's coming up, don't worry. But just so you are aware, in reality, when we actually look at most of these diseases, there are probably multiple parts of the immune system involved. So we often talk about different diseases as an antibody one or a T cell one. But in reality, for example, multiple sclerosis is usually talked about as a T cell related disease. You can see that really we see MHC and antibodies and Th1 and all sorts of things going on. And so we often classify in these different groups, and you're going to see some of that more as we go on today. However, just so you know, it's not always really as clean as that. So that was kind of the prelude I wanted to give you about autoimmunity. Hopefully it will make sense why I did it that way as I move forward. And our main topic for today, as well as for Wednesday, is hypersensitivity. Hypersensitivity is what immunologists really refer to as refer to when people more normally mean allergy. And so this is all about allergy here. That's why I got this picture. I used to do this course in the spring. I would be doing this right in the time when I was having my really bad pollen allergy season, so this was a very meaningful slide. So it hits a little differently right now. So the idea with hypersensitivity is that you are making an immune response against an innocuous antigen. So it's basically a harmless antigen. It's an antigen that you should not be making a response against. Autoimmunity is where you are making a response against a self-antigen that you shouldn't be making a response against. But in either case, you're making a response that you shouldn't be making. So one example of hypersensitivity is that you might be making a response to pollen. I make a lot of responses to pollen. I am not a tree. So it's not an autoimmune response. I'm not making a response to self, but at the same time, I'm not going to get infected by trees. It's a harmless, innocuous substance. And so that's where the dividing line really comes in here between hypersensitivity and between autoimmunity. However, there are a bunch of different kinds of similarities between them. There is also a genetic component to allergy, just like there's a genetic component to autoimmunity. In this figure, you are seeing data on genetic associations between different diseases and different chromosome locations. And so red is autoimmune diseases, which we've already talked about. And so you can see, for example, this portion on the long arm of chromosome 14 has been related or associated with autoimmunity. However, you can also see some allergies here, like atopic dermatitis, which is in the purple square. And what you can notice is that there are a number of loci that seem to come up for both the allergies and the autoimmune diseases. There are some that are unique. There are some that might be unique just to one particular disease state. But if you notice, there are an awful lot that have a fair amount of overlap between some of these different diseases. And the sort of other really important thing to realize, and this is going to be kind of a theme that we're going to see for at least this week, if not a little longer, is that when we think about hypersensitivity reactions, when we think about allergy, we classify them into 4 different types. They are cleverly called type 1, type 2, type 3, and type 4. And those 4 different types, that classification, is based on what mechanism is really important in terms of things like antibodies or T cells. And so when we think about different types of allergy, we are putting it into one of these 4 buckets. And it turns out if you understand the buckets I'm going to tell you about, those same buckets can largely be used as for classifying our autoimmune diseases. So remember how I showed you some autoimmune diseases were antibodies, some were T cells. It turns out they fall into those same types that our allergies fall into. When we later talk about transplantation, our transplantation rejection responses are going to fall into those same types. And so for the rest of the time today, as well as on Wednesday, I'm going to be talking through the hypersensitivity response types and giving you some examples and talking about how they work. But what you can realize is that by learning those general mechanisms, you are also going to be learning some details of how some autoimmune diseases work. If we have time on Wednesday, I'm going to circle back and hit the autoimmune disease stuff. And this will also help us when we get to transplantation later, because it's going to be the same exact mechanisms again. So when, Jamie, you asked about immune complexes are type 3. So we'll see it with type 3. And so really what we're going to be talking about now are the hypersensitivity reactions. And as I mentioned before, there are 4 different types. With the 4 different types, we can see differences in terms of which part of your immune system is involved in the response. So you can see immune mediator and mechanism. And so you're going to care, as we talk about these, about which part of the immune system is actually causing the problem. And just like I told you with autoimmunity, even though we usually kind of come up with, oh, it's the antibody that's the problem here, you can see that there are a few things that are probably interacting to really make that antibody response happen or something, even if we're going to blame it all on the antibody. We're also going to care a little bit about the antigen in terms of what form the antigen is in. In particular, I say that because in type 2 versus type 3, there are a bunch of things that are similar, and the big thing that's different is the type of antigen. And one other thing you may want to pay attention to here is that 2 of the 4 are special because of their timing. So in 2 of them, the timing really matters. So you'll also kind of note that timing. And so we're just going to talk through these 4, kind of what they look like, some diseases that are associated with them, all that good stuff. So the first one we're going to start with probably won't surprise you. It's type 1. And a type 1 hypersensitivity reaction is something that immunologists also know as an immediate type hypersensitivity reaction. One of the things that is sort of characteristic about a type 1 hypersensitivity reaction is its timing and how quickly it is happening following exposure to the antigen. In many cases, a type 1 hypersensitivity reaction is happening within minutes after your exposure to the antigen. You might think that I'm being a little bit picky because I'm calling all of this hypersensitivity instead of calling it allergy. One of the reasons why I'm using the words hypersensitivity here is because when people think of allergies, they think of type 1s. And really, type 2s, type 3s, and type 4s are also a thing. And most of the time, if you think of allergies, you're thinking of a type 1. So some examples, and we'll see more examples of this, of a type 1 hypersensitivity reaction, include anaphylaxis, as well as things like hay fever, asthma, hives, food allergy, eczema, just all sorts of fun stuff. All of these responses have something in common, which is how they act as part of this type 1 hypersensitivity response. So the kind of very classic aspect of type 1 hypersensitivity is shown on the left. So with a type 1 hypersensitivity response, we have a mast cell that is full of granules. And that mast cell has an Fc epsilon receptor on its surface. An Fc epsilon receptor binds IgE. Epsilon and E. Epsilon is the Greek letter for E. So what will happen is that all of the IgE in your body right now is bound to the surface of mast cells. If the antigen comes along, it will bring together a couple of those Fc receptors because of the antibody that's on them. It will basically cross-link. That will turn on this mast cell, and the mast cell will release its granules. The mast cell doesn't need to do any transcription. The mast cell doesn't need to do any translation. All the mast cell has to do is throw its granules out. And that's what allows this reaction to be immediate type, is that you are, again, within minutes, you are going to have the response here because it's going to be the response to all of these types of molecules, like histamine, as I'll show you later, that is coming out of that mast cell. So that's how we get this immediacy of this immediate type response. But there is a little bit of a caveat to the idea of this as an immediate type response. It's defined as an immediate type hypersensitivity response. If you look on old exams or something like that and you see little scenarios where I'm describing a response and I mean it to be a type 1, it's usually like, 10 minutes later, this terrible thing happened. It's immediate. But like I said, there's a caveat. And the caveat is shown here. When you are having an immediate type hypersensitivity response, it is not the first time you've ever seen the antigen. You have the immediate response the second and later times you see the antigen. The first time you have the antigen, you have a response that is shown at the top, which is a response known as the sensitization response. And so here you can see this person's first exposure to pollen. The first time this person is exposed to pollen, that's going to lead to the activation of some T cells. Those T cells are going, and remember, type 1 hypersensitivity is mediated by IgE antibodies. So if I were to list what is the mediator, IgE antibodies would be my answer. Yeah, that first time through, that antigen has to actually stimulate some T cells. Why does it have to stimulate T cells? Yeah, Sebastian. Yeah, you're not going to get IgE. You don't have IgE at the beginning. You don't have IgE before you've seen the microbe. You have some B cells making IgE. But if you're IgE, you've got a class switch. You've got to get T cells involved to help those B cells, to provide some cytokines, to make those B cells class switch. So the first time you see the antigen, you're going to activate the T cells are going to activate the B cells are going to make IgE, the IgE is going to get stuck to the mast cells, and you're going to end up in a situation like this, where you have these mast cells covered with IgE around your body. Only the second time that you are exposed to that same antigen, are you going to have those mast cells ready to respond immediately. And so the second time you see this antigen, the third time, the fourth. I don't know how many times I've seen that pollen now. The too many time. You're going to have your mast cells release, and you're going to have this immediate response. But that only happens after you've been sensitized previously. This response happens because of all of those products that the mast cells are making. And so you can see those products from the mast cells right now. The most famous of them is this small molecule here, which is histamine. Histamine, let's see here. Histamine is going to increase vascular permeability and cause muscle contraction. So basically, you're going to start an inflammatory response because you're changing vascular permeability. You're also going to have a number of enzymes that are made. Those enzymes are actually going to break down tissue matrix. That's going to help cells get in. It's also going to help with repair. You're going to have a lot of cytokines that are going to be made, although those cytokines are going to take more time. And you're also going to have a number of different lipids that are being made, specifically the leukotrienes, sometimes also referred to as leukotrienes. I've heard people pronounce it both ways. You can see these small molecules made here. This includes also things like prostaglandin. You can see a figure that shows you the sort of synthesis pathway of some of these leukotrienes from a molecule called arachidonic acid. You can see that some of these are going to do things like attract leukocytes, give you muscle contraction, give you vascular permeability, give you muscle mucus secretion, all sorts of things you think of as inflammation, right? That sounds like inflammation. You will notice on the left that if you're going to go from arachidonic acid to prostaglandin D2, it's called the cyclooxygenase pathway. Cyclooxygenase is an enzyme. If you were going to make an acronym for cyclooxygenase, you might call it COX. Cyclooxygenase is also known as COX-2, and it is one of the main inhibitors of aspirin. Aspirin is one of the main inhibitors of COX-2. So aspirin is specifically hitting this COX-2 pathway and stopping these lipid mediators. So in fact, that's the reason why I even mentioned them, is because you've probably inhibited them in your life at some point or other. And these mast cell products are going to do some of the things that you see here. So we're going to have vasodilation, vascular leak. We're going to have inflammation. We're going to have some tissue damage, both to try to give us some wound healing as well as to allow in more cells. We're also going to have a lot of smooth muscle contraction. That's going to involve or result in things like bronchoconstriction and mobility of the intestine, which is a pleasant way of talking about all of the things in your intestine coming out real fast. And so in the end, that muscle contraction in the respiratory tract is going to be that contraction of smooth muscle, which will constrict the throat and airways, giving you difficulty swallowing, difficulty breathing, and wheezing. That same effect on the muscles in the GI tract will give you stomach cramps, vomiting, fluid outflow into the gut, and diarrhea. So all of these are the very exciting results of mast cell activation. There also are some, and these are all the immediate effects of mast cell activation. There are also some later effects of mast cell activation. So these are all the things that mast cells do that happen right away. Mast cells can also have some actions that take a little bit longer that will eventually lead especially to some tissue changes and remodeling as well as some inflammation. And so there are going to be some of those factors that are going to act later and some that are going to act earlier. And so usually we think of the sort of the vascular smooth muscle response with the histamines as happening right away. So you're going to open up the vessels. You're probably going to get swelling. You're going to get this little bitty swollen spot, right? Later on, you're going to make a lot more cytokines and get more inflammation, and then you're going to have kind of that red, swollen, painful area a little bit later on. And so the immediate phase is where this all really starts, but then it can go into a late phase as well. So we can also think a little bit about why some of this happens. And just like with autoimmunity, we think that there's a role for genetics and there's a role for environmental factors. So some of this has to do with the genetics of your immune system. Others have to do with things that may predispose you in your environment, whether or not you have your barrier immune responses broken, all sorts of things like that. In terms of the genetics piece to this, we do know that some people are what are known as atopic or have something called atopy. Because my life, I I actually had to define this for someone over Thanksgiving break. Like, what's this word mean? But they were pronouncing it wrong, and I couldn't figure out what word they were saying for a while. And finally we got there, and I was like, oh, I know that word. And so it's atopy. And it means having a genetic predisposition towards making a type 1 hypersensitivity response. So basically, whenever you have an antigen, you seem to make a type 2, a Th2 response. In general, that probably has something to do with genetic susceptibility and something happened in your environment, though those parts we can only hypothesize at. But in the end, individuals who are atopic tend to have atopic asthma, atopic dermatitis, so some kind of skin issues, as well as rhinitis or a lot of respiratory allergies. And so these individuals, atopic individuals, will have all of these symptoms together. It just so happens that many of the super atopic individuals, weirdly enough, also have extraordinarily pale skin. It's weird. I am the poster child for atopy. And surprising to no one, I am super atopic. And so there are some people who do have this genetic predisposition. And when we look a little bit at some of the genes that are involved in type 1 hypersensitivity, you can see that they seem to be related to things like epithelial barrier integrity. We've got some PRRs. We've got some chemokines and cytokines. We've got some things involved in tolerance. But we also have some things, at least in the case of asthma, which is shown on the right, that are just genes involved in what your airway structure is like. And so there's a little bit of sort of both pieces of this here. The type 1 hypersensitivity responses can be a lot of things. And so these are some of the allergens that we see that can be related to a type 1 hypersensitivity response. They include all the fabulous pollens. They include most of our big food allergies. They include the animal dander allergies, mold allergies, latex allergies. This includes a fair number of drug allergies. This also involves allergies to insect products. So this is a bee sting allergy. This is also a dust allergy. Most people don't know this. It's always fun and exciting. So when you're allergic to dust, the thing you're actually allergic to is something called a dust mite, which is a little tiny arthropod that you can't see. It looks like that eats the dust. And it actually has a protease, and you're actually responding to a protease from this little microscopic arthropod. You're not actually responding to the dust at all. And so dust mites and other insect products can be a big part of this, along with insect venom. Some metal allergies, some plant oils, all will be a problem here. This lists penicillin. There are actually examples of penicillin allergies in all 4 types of hypersensitivity responses. I'm going to talk about one of the other penicillin allergies, not a type 1, later on. And if you look at the responses to all of the things that are shown here, you might say, yeah, Dr. Barker, those are some pretty different responses. I know people with allergies to those things. It's hard for you. I'm not sure I can believe you that they're all the same process. And really, what's going on here is that these are all type 1 hypersensitivity reactions, but they're happening in different anatomic locations. And so a type 1 hypersensitivity reaction looks a little bit different when it happens in a different anatomic location. And so we can see one kind of response that happens when we're having the antigen come in through the skin subcutaneously. A different kind of response happens if you inhale. A different kind happens if you have that response coming in through the mouth and into the GI tract. And yet another one happens if you actually have the same antigen coming in into the blood. And so basically all of these different responses, while they look really different, are the exact same thing happening, just at different anatomic locations. So this is that process happening in the skin, the type 1 hypersensitivity in the skin. It's sometimes known as the wheel and flare. And you can see examples of this here. So you have mast cells that live in the skin. So you can see those mast cells are living in the skin. And if we have antigen exposure, the mast cells in the skin will degranulate. That will lead to changes in just the blood vessels near the skin. And those blood vessels will get leaky, and we'll have fluid, cells, and proteins coming in at just that area. And so you'll get a little swollen spot right there. That may also be itchy because some of those may impact nerves. And you're going to get sort of that characteristic allergic reaction. This is an example of a patch test. If you go and say that you think you have an allergy, and you go to your doctor's office to get tested, your doctor will give you a patch test. And so the idea is when those antibodies, when those IgE antibodies that you have made bind antigen, they're not going to like only bind antigen in one place. They're going to bind antigen throughout your body. Sorry, they're only going to bind mast cells throughout your body. But they're only ever going to get triggered in the location where you see that antigen. And out of all the responses I'm going to show you, this response in the skin, the wheel and flare reaction, is not the most serious. Some of the other ones are really dangerous. So what we do is we just expose you to a whole bunch of antigens under the skin with the idea that the mast cells of the skin will have this, those IgE antibodies, and we can see if you make a response in this kind of safe way. And so we can just see what you're allergic to and what you're not. So this is an example. Here the person got a negative control, and you see that nothing happened. They had no wheel and flare. Here they got histamine, positive control. That's the response. Here they got plain pollen. I don't know what plain pollen is. Here they got birch pollen, here they got grass pollen, here they got daisy pollen, they got mold, sheep wool, horse, dog, cat, feather. And so you can see all the things that this person is allergic to. In some cases, you actually make such large responses, they start to bleed into each other and it becomes really hard to see them. If that is the case, what they actually do is they repeat the patch test on your back. So they have more surface area to see the full responses, because that sounds lovely. Yes?
Is it possible that you might not have a response to this test, but it could be like your first exposure to one of the antigens, and then you have a response the second time you're exposed?
In theory, maybe. I don't know of examples of that, but theoretically that could happen. But yeah, so this is also how you get, this is a lovely example of what a patch test, of what a wheel and flare reaction looks like, and it's how we test for these allergic reactions. If we make the exact same response in the respiratory tract, now we have those mast cells in the respiratory tract responding to inhaled antigen. That mast cell activation is going to change the blood vessels. So we're going to see vascular permeability and activation of the epithelium. But we're now also in a location that has some smooth muscle. Your skin doesn't really have as much muscle, have the same kind of smooth muscle. So now we can start to get the muscular changes. We're also now in an anatomic location that has mucus. And so we can also get a whole bunch of mucus production. So we're going to start to see mucus secretion in the airways. We're going to see decreased diameter of the airways. And this is going to result in expulsion of airway contents through coughing, sneezing, and expulsion of phlegm. We're also going to see that smooth muscle constricting in the airway, making it hard to swallow, breathe, and making you wheeze. And so it's the same process of those mast cells getting triggered. It's just that the location around the mast cells, the location that is responding to the mast cells, has different features and has a different kind of response. These are also the same kind of steps that will happen in asthma. You can see the early and late responses in asthma here. So this is what the bronchus should usually look like. You can see kind of the thickness of this layer is that it's not very thick. You can see that we've got kind of some cells in that layer, but maybe not tons. In the early response, we're going to have vasodilation, bronchoconstriction, and mucus secretion. However, we're also going to have some cytokines that are coming to be made. We're going to bring in some other white blood cells. If this happens to you over and over again, your airway is going to get full of mucus from all this mucus being made. You're also going to get changes to the wall of the bronchus, and that's because of those enzymes that are made to do tissue remodeling. It's good in the short term because you can let in more leukocytes, but it actually changes the structure of your bronchial tubes, and they aren't elastic anymore. You can't breathe as easily. So we also have we have way too many cells in here as well. And so you can see that if this happens all the time in one location over and over and over again, that one location is going to sustain some damage that is going to lead to additional problems. We can also see the same thing happening with food allergy. It's just that now the mast cell lives in the GI tract. And when the mast cell lives in the GI tract, it will lead to changes in the blood vessels in the GI tract, as well as a lot of changes to the muscle. And so we'll get vomiting and diarrhea, as well as fluid outflow into the lumen, stomach cramps, all that stuff, because we're going to hit the muscles pretty quickly. If antigen goes into the blood vessels, it can go to some other locations, which potentially causes this fancy thing here, urticaria. Urticaria is a fancy word for hives. And that basically is a situation where you're getting a little bit of inflammation in some area of the skin because of the antigen traveling through the body, through the blood. Yep.
So then with food allergy that causes anaphylaxis, is it just from it going through the, like,
Yeah, well, anaphylaxis is coming, but yes. And so here you can see kind of some of those details of the immune basis for the food allergy reaction. And realistically, there are some people who have food allergy that shows up as a rash because the first part of it is really from you touching the food to eat it. And so in some cases, we're going to get sort of a lot of rash or issues with GI inflammation. We also can see times where really the first contact is going to happen in the mouth. And so you're going to get itching and swelling in the mouth because you're triggering mast cells in the mouth. You don't even have to degrade the food for the antigen to start hitting mast cells. Again, you can see skin contact. If it actually gets further into the GI tract, then you're going to get the nausea, vomiting, and intestinal pain. And so you can see that there is a big variation here. Yep. Uh-huh. Uh-huh. We would love to know the answer to that. We would love to know the answer to that. It is probably something about genetics, that you have some kind of genetic susceptibility, and there's something that happens early in life that alters your responsiveness to that antigen. But what exactly it is, we're not sure. So I mentioned to you guys beforehand that there is this idea out there that I think makes a lot of sense. For example, with peanut allergies, that one of the reasons why we're seeing a rising number of peanut allergies is because babies are not getting peanut antigens the first time orally anymore. They're now getting it through lotions. And so they're making a inflammatory response instead of a tolerogenic response and ending up with allergies because the first time they saw it was not through the mouth, was through the skin. So, there could be things like that going on. We don't know the answer to most of those things. We would love to know the answer to many of those things, and we just don't. Yeah?
How do you develop allergy and how do they go away? Because I know some people have late onset of that and early.
So a little bit unclear, but the basic answer I can give you is for the late onset allergy, it takes multiple exposures to get that Th2 cell and to get IgE production. So, the first exposure doesn't push you to Th2. Maybe it takes you multiple times of seeing that before you actually get enough stimulation to get to Th2, before you get enough to get to IgE, things like that. So it's going to take multiple. You could imagine that the sort of outgrowing of allergies is a situation where tolerance starts to take over, maybe Tregs starting to take over. But again, we'd love to know the answer to that. And so, you can see that all of these differences are really based on whether we're seeing that antigen through the skin or through the intestine, and that will also change the specific symptoms that we see. So you could be allergic to a food and see symptoms both from touching it in the skin or in the intestine. The final type of response is anaphylactic shock. This is where now you are actually seeing mast cells that are responding to an antigen that's in the bloodstream. And so now you're not going to have one little local location and one little local blood vessel that is acting. You're going to be messing with blood vessels throughout the body. You're going to have histamine going throughout the blood. And so this is going to have major impacts throughout the body. You're going to have issues with the heart and vascular system. So increased capillary permeability and the fluid going into tissues. And so tissues are going to swell. And you're going to have lots of tissue swelling. This may include the tongue, which is going to also make it really hard for you to breathe. You're going to have a loss of blood pressure because there's no more blood in your veins or in your arteries, so you don't have any blood pressure. You're not going to be able to get oxygen to your tissues. Your heart's not going to beat regularly. You may lose consciousness. And so all of that's going to happen because of this anaphylaxis happening in the blood and the antigen being in the blood. And so yes, if food allergy leads you to anaphylaxis, usually it's because the allergen got into the blood. You're going to have contraction of smooth muscle, constriction of the throat and airways, and you're going to have various GI issues. And so here you can see some of the signs of anaphylaxis or anaphylactic shock. And so this is an immediate response to having some kind of allergen in the blood and leading to blood mast cell activation. Yep.
So with anaphylaxis
Uh-huh.
in the blood,
Yep.
why does it not, I mean, I know it leads to hives, but why does it specifically cause swelling of the respiratory tract? Is it just because that's the main tubular thing in the body?
It's because it is a place where there's a lot of smooth muscle that that is very responsive to many of these mediators and can respond easily. In addition, the lungs filter, trap a lot of things. Like if you do an IV intravenous injection and put something in the blood, it ends up getting trapped in the lungs pretty quickly. And so anything coming through the blood is going to hit the lung pretty quickly. And the lung is especially responsive to these molecules. As you might know, that I think allergies are not very great. And so we might wonder a little bit about why the heck allergies evolved. Like, why do we need any of this response? This response seems dumb. I think this response is dumb. Mostly in the spring, I think it's awfully dumb. However, what we realize is that most of our allergens are actually very cross-reactive with things that are pathogens. So if we look at a number of different helminth antigens, they are actually very similar to many of our other types of allergens. Some of these other things from some of our other helminths are very similar to some of the other allergens. And so we know that this response, this Th2 response leading to eosinophil and mast cell degranulation, is really, really important for trying to kill extracellular pathogens like helminths. And so some of this is, it just so happens that these things that we are allergic to cross-react with helminths, and so we're making the right response to get rid of a helminth. It's just, unfortunately, we're not actually infected with a helminth. And so we're kind of making this inappropriate response due to cross-reactivity. And this is sort of the generally accepted reasoning on why we have allergy. There is one other hypothesis that is out there, and I just want to kind of point out this hypothesis because I do think it's kind of interesting. If you think about having a helminth infection, think about having a tapeworm. Is there anything that's super important to you about speed in dealing with that tapeworm? Is it like that important to like deal with a tapeworm fast? Yeah. Okay, size is important. Does it matter how fast you are? Do you have to be like crazy fast to deal with that tapeworm? Not really. Like, you don't have to be faster than any other type of response, right? You have to be faster to deal with a tapeworm than a bacteria. Like, yeah, these responses, these immediate type hypersensitivity responses are immediate. They're happening within minutes. They seem to have evolved to happen so fast. And it's kind of like, why is it the tapeworm response fast? Like, what? It doesn't make sense. Yeah?
Is it because we want to get rid of the tapeworm before it reproduces?
It doesn't take it reproduces real slow. And so there is this alternative hypothesis that also talks about the fact that it also seems like an awful lot of these allergens are cross-reactive with venom. And so if you think about it, so first of all, let's remember the difference between venom and something that is venomous and something that is poisonous. What does it mean if something is poisonous? You drink it and you die. If I had a poisonous snake, that would mean if I ate the snake, I died. What does it mean if something is venomous? If it bites you, you die. So if the snake bites me, I die if it's venomous. If it's a poisonous snake, I have to bite the snake. It's a question of, is it bad because I ingested it, or is it bad because it gave me some bad thing? So here we're talking about venoms. And it looks like perhaps there's actually cross-reactivity with things like with different kinds of venoms and other types of irritants. And one thing that can be really interesting there is with a venom, you got to respond to it fast, right? If your venomous snake bites you, you need to get rid of that venom fast. And what do you need to do with that venom? How are you going to deal with the venom? Yeah. Get it out of your body. You want it out. You want it out fast. You want it, and if you think about most of the responses we see with allergies, the wheezing, the diarrhea, all of those types of things, they are get stuff out of body fast types of responses. And so you could also imagine, that's maybe not super helpful for a helminth, but for venoms, that could actually be really key. And so there is also some more discussion in the literature that maybe this is actually our anti-venom response that has gone wrong. All right, so that was a type 1 hypersensitivity response. Now we're going to move to thinking a little bit about a type 2 hypersensitivity response. I'm only going to make it partway through type 2 hypersensitivity. I've got 2 examples. I might make it through one of the 2. So with a type 2 hypersensitivity response, one thing that's really important is that we are again seeing antibodies as the main culprit. The antibody can be IgG or the antibody can be IgM. Another thing that is really important is the type of antigen here. The antigen is on the surface of a cell. So we're going to see some antigen that's on the surface of the cell that is being bound by IgG or IgM. And the thing that is going to happen in a type 2 hypersensitivity response is that the cell with the antigen on its surface is going to get killed. So we're going to have antigen on the surface of some cell, and we're going to kill that cell as a result of the antibody binding. That may be through NK cell activity and ADCC. It may be through complement-mediated lysis. But in either case, we've got antibody. It's either IgG or IgM. We've got antigen on the surface of the cell, and we're going to kill that cell. And so that's going to be sort of the big message of what happens in a type 2 hypersensitivity response. I've got 2 examples. I'm actually going to tell you the one that I think is a little bit shorter, which is the second one. I'll do the other one to start next time. And the one that I want to tell you about is the penicillin allergy that happens with type 2. You can see that you can actually get a penicillin allergy for all 4 types. But the one I'm obviously telling you about now is the type 2 response, which leads to something called hemolytic anemia. This is penicillin. Penicillin has a bond in it that is chemically very reactive. And what penicillin is supposed to do is react with this enzyme from bacteria so that it doesn't work, so the bacteria die. But sometimes that reactive bond can act against red blood cells. And so then we get red blood cells with penicillin attached, which you can see on the right. This penicillin attached to the red blood cell is now a new antigen on the surface of the red blood cells. And so you can see our red blood cell with penicillin attached could get antibodies bound to it. And this could lead to this red blood cell dying either by complement-mediated lysis, by phagocytosis, or actually NK cells could kill as well. And so now this person who has this happen is going to lose their red blood cells and is going to have anemia as a result of autoimmune attack on these red blood cells, which are coated with penicillin, which becomes a new antigen. The antigen is kind of like the bonded at the bond site there. And so that's one major example. Again, notice we've got a cell surface antigen. We've got antibodies, either IgG or IgM, and we're killing our cell. And we'll see the other example of this next time, and then we'll go into type 3s. So I will see you guys on Wednesday.